Welcome students. In today's class, we are going to see the food journey in the second part of the digestive system, that is from stomach onwards. And we will see what are the different enzymes that are produced in the different parts of the digestive system and how the process of digestion and absorption is going on. So after the food has entered into the esophagus and it has come into the next part to which the esophagus is connected is the stomach. And in the stomach, the digestion of protein begins. And in the stomach, let us first take a close look at how the stomach looks like. The stomach actually looks like a sac. It is more of a sac-like structure. At one end, it is attached to the esophagus, and at another end, it is attached with the duodenum part of the small intestine. Now, the entrance of the esophagus to the stomach is guarded by a sphincter, which acts like a gate, which is called the cardiac sphincter, and the entrance of the stomach to the small intestine or duodenum is guarded by another sphincter, which is called the pyloric sphincter. Now, these two sphincters are very important in maintaining a single flow or unidirectional flow of the food. So, the cardiac sphincter is only opening in the direction of the stomach so that the food can only pass from esophagus to the stomach and not in the backward direction, that is from stomach to the esophagus. And similarly, the pyloric sphincter is also opening in only one direction so that the food can only pass from the stomach to the small intestine and not in the backward direction. So these two sphincters are guarding the opening and exit of the stomach for preventing the backflow of the food. But when vomiting happens, that is, the stomach is overloaded with food material and acid or something. So here only backflow of the food happens, where the cardiac sphincter will open and antiperistalsis will occur in the walls of the esophagus, and we will expel the excess amount of food that was causing the discomfort. But it only occurs when we are vomiting and never else. After seeing the structure of the stomach, now we will see the process of chemical digestion of food in the stomach. The inner lining of the stomach has some cells or glands that are secreting a gastric juice. This gastric juice is actually a combination of different things. It is having HCl in a lot of amount, which is an acid, pepsin, which is an enzyme, water, and some of the salts. Now the HCl is actually killing the germs that are present in the food. Saliva was also acting as anti-germ solution or anti-germ solvent, but still some of the germs might also be present even after the action of the saliva. Those germs will be killed by the HCl in the stomach. And the pepsin, which is the enzyme, it is secreted in an inactive form inside the stomach, and HCl is having a major role in activating that pepsin from its inactive form to the active form. Pepsinogen is the inactive form of pepsin. HCl is acting on pepsinogen to convert it into the active pepsin. Now the activated pepsin is acting on proteins to break it into smaller parts or simpler parts of protein, which are called peptides. The prorenin is also an inactive form of the enzyme renin. HCl is also acting on prorenin to convert it into the active renin. The activated renin is then again acting on the milk protein, which is the casein, to turn it into smaller or soluble fragments, which are called paracasein. So these are the two chemical reactions that are taking place in the stomach, but all of them need HCl because the enzymes secreted are in inactive form. As as the food mixed with saliva was termed as bolus, but when it has come into the stomach, it has also get mixed into gastric juice. So the food after digestion into the stomach, that is the food mass that is going into the small intestine mixed with the gastric juice, is called a chyme. The next destination after the stomach that the food goes into is the small intestine. So it is the most coiled portion of our digestive tract. As we can see in the picture, from the base of the stomach, the duodenum or the first part of the small intestine is originating. It is the short upper part just next to the stomach. A common bile duct is opening here from the liver. We will see that in detail in later slides. And after the duodenum has traveled a little bit distance, the tubes or the pipes of the small intestine get coiled. And those coiled portion, the upper portion of the coiled part is called jejunum and the lower part is called the ilium. And it is the longest part of the small intestine out of the three parts. And in ilium, the inner lining is having some finger-like projections, or inner wall of the ilium is having some finger-like projections which are called villi. And these villi are having a lot of role in absorption of the food. Now we will see what are the digestive processes or chemical processes that are taking place in the small intestine. Now it is a little bit complicated and elaborated because most of the digestion is taking place in small intestine. So there are a lot of enzymes involved in small intestine as well. Now as we are seeing, from the stomach, the acidic chyme mixed with the gastric juice is coming into the small intestine, and also from the walls of the small intestine, intestinal juices are secreted. From the pancreas, as we can see, there is a pancreatic duct opening into the duodenum as well. So the pancreatic juice from the pancreas is also coming into the small intestine, and also we can see that from the liver and the gallbladder, the bile is also coming into the small intestine. So there are three chemicals that are actually going to work on the acidic chyme that has entered into the small intestine: the pancreatic juice, the intestinal juice, and the bile. First, let us take a look at the pancreatic juice and what are the components present in the pancreatic juice. First is another enzyme that is present in pancreatic juice, which is called amylopsin. Amylopsin is actually working on carbohydrate or starch and turning it into maltose. It is also having another enzyme which is called trypsin, and it is acting on proteins to turn them into peptides and amino acids. So peptides and amino acids are even simpler and smaller fragments than peptones. These peptides and amino acids are going to get absorbed in our blood. Stepsin is another enzyme that is present in pancreatic juice. It is working on the fat to turn them into fatty acid and glycerol. So those are actually the smaller or simpler fragments of fat, fatty acid and glycerol. So the pancreatic juice is having three types of enzymes which are acting on starch or carbohydrate, protein and fat. Now we will read about the intestinal juice and what are the enzymes present in it. First is the erepsin. Erepsin is an enzyme that is working on the peptides to turn them into amino acids. So erepsin does not work on the proteins, but it is actually working on farther segmented or farther fragmented portions of protein, which is the peptide, to turn them into even simpler form of protein, which is amino acid. And also it is having maltase. Maltase is also an enzyme which is converting maltose into glucose. Glucose is the absorbable form of carbohydrate that can be used by our body. It is having lactase, which is converting the lactose like present in milk into glucose and galactose. It is also having sucrase, which is converting sucrose into glucose and fructose. And it is also having lipase, which is acting on the fats to turn them into fatty acid and glycerol. And some of the absorption is also taking place through the intestine. Amino acids and sugars are actually absorbed through the epithelium of villi. Villi we will see in the next slides. And fatty acid and glycerols are absorbed into the lymph vessels that are present in the intestinal walls. The bile is actually a secretion that is secreted from the liver. Then from the liver it is coming to the gallbladder where it is stored. So the gallbladder is not actually secreting the bile, it is storing the bile and it is concentrating the bile. That is getting rid of the water content from the bile and making it very concentrated. So the hepatic duct is the duct that is coming from the liver. Cystic duct is the duct that is coming from the gallbladder, and together they are forming the common bile duct which is opening into the duodenum. Now in the bile, what is there? Bile is actually a yellowish green watery kind of fluid, and it is having two proteins which are called bilirubin and biliverdin. Now bilirubin and biliverdin are actually produced from dead RBC or dead red blood cells. Bilirubin and biliverdin are actually yellowish in color, and those bilirubin and biliverdin are actually present in the bile as the main component. And also bile is having some sodium bicarbonate as neutralizer. What is a neutralizer? The food particle or the chyme that is coming from the stomach to the small intestine, it is actually containing a lot of HCl that was produced in the stomach. So those HCl is actually very harmful for the intestinal wall. It can actually corrode or do harm to the intestinal wall. So to neutralize that acid, sodium bicarbonate is produced by the bile so that the acid of the food particles cannot do harm to the intestinal wall. And bile is having the main role in emulsification of fat. Emulsification of fat means the bile actually segments the fats into smaller droplets. So the surface area is increased and more enzymes can act on the fat droplets or fat molecules so that they can be turned into fatty acid and glycerol, which can be used by our body. Small intestine is the first portion where, apart from digestion, absorption of food is also taking place. So in the previous segments of the alimentary canal, no absorption was taking place. Only digestion was taking place. But in the intestine, in the ileum mainly, the absorption of food is also taking place. Because the inner wall of the ileum is actually having some finger-like projections which are called intestinal villi. So it is actually increasing the surface area for more efficient food production, food absorption. Now intestinal juices are poured through the small pores of the villi in the intestinal lumen. So there are some pores present, or there are some places present inside the villi through which the intestinal juices are getting inside the intestinal lumen. And as we have seen that intestinal juices are actually having so many enzymes that are digesting the food. Now the villi is having an outer surface which is a single cell thick epithelial layer. As we have already studied in the previous chapter, that epithelial layer is a sheet of cells. So as we can see in the picture as well, so there is an outer covering of the villi which is a single cell thick epithelial layer, and inside that there are the blood vessels that are present and also veins present, artery present, which are part of the blood vessel, and lymphatic vessels present. Because some of the food particles will be absorbed into the blood and some of the food particles will be absorbed into the lymph vessel. So lacteals are nothing but the lymph vessels. The bile and the pancreatic juice in the duodenum and intestinal juice in the ileum, these are actually the digesting juices. And after the digestion process is completed, the villi is going to absorb those simple food particles into the blood and the lymph. After the digestion and absorption has taken place in the last portion of the small intestine, that is the ileum, the undigested food particles are going to move into the large intestine, which is the last part of the digestive tract. So there are three portions of the large intestine: cecum, colon and rectum. Cecum is the pouch at the junction of small intestine at the large intestine. That is the left hand side portion, as you can see. It's just the junction. And it's a small pouch like portion. It's a little bit bulged portion. There is one part at its end which is called the vermiform appendix. And vermiform appendix is a vestigial organ present in our body. That is, it is having no purpose or no function in our body. The colon is the next part of the large intestine and it is the most largest part of the large intestine. Colon is having three parts: the ascending colon, which is starting just after the cecum. Then there is the transverse colon and then there is the descending colon. And at the end part of the descending colon, it is called the sigmoid colon. And sigmoid colon is actually ending into the rectum. Rectum is the last part opening into the anus. At the opening of the anus or anal canal, there are circular muscles present that help us to defecate whenever we want. The large intestine is the part which is producing no enzyme. So no digestion is taking place. So last part in which the digestion has taken place was the small intestine. But colon has another important part in digestion, which is it is absorbing the water from the digested parts of the food so that it is actually a little bit solidified. It is having or storing mainly undigested food, and since it already has absorbed water from the undigested food material, it is forming semi-solid feces. It is helping in the defecation process, that is helping us to expel the undigested food materials from our body. The bad odor in feces is due to the action of the bacteria that are present in the colon. So those bacteria are actually acting on the feces to turn them into more smaller parts so that they can be just mixed with the soil when we are defecating. After the food particles are absorbed by the blood vessel and the lymph vessel, through the blood and through the lymph, they are going to different organs of our body, different organ cells. So they are going to our brain cells, our heart cells, our liver cells, lung cells, our body skin cells and everywhere. Now these body cells are actually converting those absorbed food material into body material. That is, they are using it for some purposes. This using process is actually called assimilation. Two systems are responsible for this assimilation process. One is hepatic portal system and another is lymphatic system. Hepatic portal system is the blood system or blood circulation that is entering into the liver. The liver is very important in assimilation because liver converts the excess glucose into glycogen. The glucose that is coming to the liver through the hepatic portal veins, liver is converting that glucose into glycogen for further storage, for future use. This process is called glycogenesis. And when our body is needing that glycogen, so liver is actually converting that glycogen into glucose by the process glycogenolysis. Lysis means breaking. So lysis means breaking of glycogen into glucose. so that glucose can be used by our body again. Liver also breaks down the excess amino acids, which is called deamination. Fatty acids and the glycerols are transported through the villi and the lymphatic system. So they are absorbed by our body as well through the villi and the lymphatic system or the lymph vessels.